I'm very pleased to introduce Dr. Mark Gluck, who's a colleague of mine at Stanford University in the cognitive psychology program. Having left Stanford, I went off to computer science, and Mark went off to further academic efforts. But both of us are interested and have been interested in the way the mind works, and hopefully Mark will give you some insights into what he has been doing over the last few years. Mark is a professor of neuroscience at Rutgers in Newark, and he's co-director of the Rutgers Memory Disorders Project. He's also published a newsletter, Memory Loss in the Brain, and he works in the interface between neuroscience, psychology, and computer science, where his research focuses on the neural bases of learning and memory. He is the co-author of Gateway to Memory, an Introduction to Neural Network Models of the Hippocampus in Memory. I'm sure you've all read that. And he also is about to come out with an undergraduate textbook, Learning and Memory from Brain to Behavior. He has won a number of awards, including the Distinguished Scientific Award for Early Career Contributions from the American Psychological Society and the Young Investigator Award for Cognitive and Neural Sciences from the Office of Naval Research. Mark has also done some really interesting work on, he's told me about years ago about detecting potential flaws in helicopters and other sort of interesting things. So it'll be interesting to see what he tells us about today. I'd like to introduce Dr. Mark Gluck.
Thank you. Thank you. So can everybody hear me with the mic? So what I want to talk about today is some of the work in our lab at Rutgers that, as Eddie said, is at the intersection between psychology, neuroscience, and computer science. And I'm going to focus on how people learn associations, how they learn to categorize the world, and particularly how they learn to generalize what they've learned in one situation to other domains. And the most of the talk, I'll talk about the sort of applications to the biology and the psychology, trying to understand how the human brain does these and how we can infer the algorithms that are used to do this. And then in the last part of the talk, briefly touch on some of the applied problems in machine learning and pattern recognition that we've worked on, and hope that perhaps some of these applied aspects of our work might touch on some issues of interest to the people here at Google. And I'd love to sort of interact and talk with people afterwards about how some of the things that we're doing might be relevant to you. So part of the core of what we study is how do people categorize what they know in the world? How does a student of Impressionist art learn to tell the difference between a Monet and a Manet? And there may not be any simple rule, but you can begin to develop associations between different clues that suggest one category or the other, so that one might learn that this image should be categorized as a Monet and that image as a Manet. And we're interested in how do people form these kinds of categories, how do they learn from exemplars, and how do they then generalize this to novel instances. I'm going to talk actually primarily about two brain regions here. The basal ganglia, and argue that it's critically involved in how we learn about feedback from the environment about the consequences of our behavior. When we make an action, we get some feedback from the environment, positive or negative, and it influences our future behavior. And that, we've argued, is critically involved with the basal ganglia. And then, that'll be sort of the first half of the talk. In the second half of the talk, I'll talk about the hippocampus, which we've argued is critically involved in determining new stimulus representations for information that comes in. And those new stimulus representations are, the encoding scheme depends on detecting regularities in the environment. And these two structures work together, one to develop how to represent information, the other to use those representations in order to sort of change our likelihood of making one action or the other. And I probably won't talk much about the frontal lobes in this because I'm trying to keep it, the talk shorter. So the approach that we take in our lab is multidisciplinary. We use functional brain imaging, patients with focal brain damage, genetics, comparative studies of lesioned and transgenic animals, and neurocomputational modeling. And so to bring these all together to provide some converging evidence for what these different brain regions are doing in the context of learning about concepts and categories in the world. So the first question that I want to start with is a psychological one, or a philosophical one, going back even further. What are the principles which govern the learning of new associations? So the earliest writing on this traced back to Aristotle, who described two principles. One of contiguity. When two things are close together in time or space, we tend to associate them. And when they tend to co-occur, we tend to associate them. And these principles still today sort of define much of what we know about when it is that we tend to associate one thing with another. And these principles of association led in the mid-50s in mathematical psychology to the development of try to develop algorithms to describe how animals and people learn. And the approach here was to try to come up with mathematical equations which would take information about the contiguity and the frequency and other factors and develop quantitative predictions for behavior and learning. And Clark Hull was sort of the father of this. And although Hull's work itself is not really relevant anymore, his work led to some very important work in the late 60s and early 70s by Rescorla and Wagner, who developed what we call an error correction model of classical conditioning, of basic associative learning. And they had two key ideas. And the first idea they had was that in addition to the contiguity and the frequency, that what's critical is that cues provide useful and reliable predictive information about expected outcomes. That it's not just enough for two things to co-occur, but one of them has to be a reliable and useful predictor of the other. And they argued that the way that animals learn this, even the most simplest of animals learn this, is through trial-by-trial changes in weights that correct or seek to minimize prediction errors. And the prediction error is the difference between what an animal expects and what it actually occurs. It's, in a sense, a measure of surprise. And the basic principle of error correction learning characterizes three different situations that are of interest. One is if one has a cue and it predicts nothing, it's a neutral cue, and something occurs, then we have a positive prediction error. And the Rescorla-Wagner model says you want to increase the associative weights. If you have a cue that predicts an outcome and the outcome occurs, then there's no prediction error, there's no surprise, and you leave everything the same. And finally, if the cue predicts an outcome and it doesn't occur, you're disappointed, then you have a negative prediction error and you decrease the weights. And for those of you familiar with neural networks, this is basically the least mean square algorithm for minimizing expected error over a set of patterns of inputs and outputs. The Woodrow-Hoff rule, in fact, is what it's called in engineering. So here's an example of how we study error correction learning in people. We teach them to be tarot card readers, gypsy fortune tellers, and we present them with many trials that have patterns of cues like this one. And we say, do you think this predicts rain or sunshine tomorrow? And they'll push a button, rain in this case. And it turns out it is a rainy day, and so they get positive feedback. And then from this learning of this pattern, they may see another pattern with other cues and think, well, maybe this also predicts a rainy day, but in fact, it's a sunny day. And we give them many, many trials like this, 50, 100, sometimes even 200 trials. And the way in which the experiment is set up is that there are 4 different cards which can occur on any trial, and they have probabilistic relationships to different outcomes. So the card with the squares at the top is associated 20% of the time with sunshine and 80% of the time with rain. And what we're looking at is how do people learn these sorts of probabilistic rules, these associations between cues and outcomes. And back when Eddie and I were graduate students at Stanford, working with Gordon Bauer there, Gordon and I had argued that the way people learn this is fundamentally the same as the way that animals learn classical conditioning, like Pavlov's dog learning that cues predict food. And that one could capture this at an algorithmic level in a simple connectionist or neural network that associates the cues as well as combinations of the cues with the different outcomes, good weather and bad weather. And that trial by trial, the ways that people adjust their underlying knowledge of these categories is through this kind of error correction procedure that's found in animal conditioning. So that the learning on any trial is governed by some learning rate, beta, times the prediction error, the difference between the actual category, rain or sunshine, and the degree to which the person expects these two different categories. And we showed that at a psychological level, one could account for a wide range of data on people's choice behavior and probability estimates and so forth. So this formed the background for some of our current work, and let me just give you a brief interim summary of what I've discussed so far. So 1, contiguity and frequency are key to associative learning. 2, that cue outcome learning only occurs when a cue fails to predict an outcome, and that the association weights change to correct the prediction error. And 3, this same error correction principle that was originally developed as studies of animal conditioning also apply to how humans learn probabilistically defined categories. So now let me turn to the basal ganglia brain structure, which I've argued is critical for how we learn from feedback about our actions. So the basal ganglia are shown here, and what's critical about the basal ganglia is that they are modulated by projections from deep in the midbrain, which send dopamine, a neuromodulator, which is critical for the functioning of the basal ganglia. And the reason this is of particular relevance is that there have been some studies of these dopamine cells, in monkeys, and I'm going to show them to you right now, and you'll see why they're relevant. So in work done by Wolfram Schultz and others, they showed that if you record from these dopamine cells, these cells which project dopamine to the basal ganglia, during conditioning where a monkey is being trained that different cues do or don't predict a juice reward, if you look at the outlined in sort of the red dotted line that shows what those cells are doing at the point where the juice either does or doesn't occur, and what they found is that those cells are firing in proportion to the error signal, the error term, the prediction error from this Rescorla-Wagner model. So when the monkey does not expect the juice and the juice occurs, there's a positive prediction error and the dopamine cells fire. When the monkey is well trained that the cue predicts the juice and it expects it, there's no firing. And if you look, you can see it's a little bit subtle. When the monkey expects the juice and there's no juice presented, you'll see there's a dip in firing. There's normally a baseline level of firing in these dopamine cells, and when the animal is disappointed, it shuts down a little bit. So we see here that there is this mapping between these algorithms, these error correction learning algorithms during learning, and what these dopamine cells are doing in the brain. So this work was all done in monkeys, and our interest was how does this help inform our understanding of how people learn more complex cognitive categories. And one approach is to look at patients with Parkinson's disease. And Parkinson's disease, although it's primarily known as a motor disorder, it actually also has cognitive components as well. And what happens in Parkinson's disease is that these cells in the substantia nigra, one of the regions that's projecting dopamine to the basal ganglia, die off. And in fact, by the time someone first presents with the symptoms of Parkinson's, the tremors and the shaking and the stiffness, they've lost about 70 to 90% of these dopamine cells. So it gives you an idea of just how much redundancy is built into the system. So our first expectation was, given that these people, Parkinson's patients, have a loss of these error cells, we might expect them to be impaired at learning from error-correcting feedback. And that's exactly what we see when we train Parkinson's patients compared to healthy controls on this weather prediction task. We see that they are impaired. And that's exactly what you would expect if in these sort of error correction models, if rather than a full error signal, you have a significantly reduced error signal because these error neurons have lost about 90% of them. So another approach is to look at functional brain imaging, to put people in a magnet and have them do the task while solving this problem. And what we found is the following. Is that early in training, the medial temporal lobe, which is a brain region I'll talk about later, starts out high degree of activity and drops off. And the basal ganglia, I can't see it so well in the light green, the basal ganglia, a particular region of it called the caudate nucleus, starts out low and increases. So as the person is learning this task, the basal ganglia are growing stronger and stronger, with exactly what we would expect if the basal ganglia is critically involved in this sort of learning from feedback. We've also looked at trial by trial in the magnet at the trials where people are either getting positive feedback or the trials where they're getting negative feedback. So where they've either made a correct response or an incorrect response, and asked where in the brain is the activity the most different on those two different types of trials, subtracting the trials where people got it correct from the trials where they got it wrong. And the idea where there would be the biggest difference is where you'd expect the brain to be encoding the error between positive error and no error. And in fact, indeed, it is these midbrain, this area of these dopamine cells, which is where in the brain you see the biggest difference between negative and positive feedback trials. So two different ways of sort of looking at the brain imaging evidence of the role of the basal ganglia and these dopamine regions. Another approach is to look at the genetics. We've looked in schizophrenic patients at a particular gene that controls dopamine function in the brain, and there's a bad version of this gene called the S allele, which leads to impaired dopamine function. And what we see is that in both the healthy controls as well as in schizophrenic patients, they're slower. So even forgetting the fact that they're impaired, even among healthy controls, it's how you have this dopamine gene that determines to some degree your performance. So, brief interim summary, we've seen evidence from clinical studies of Parkinson's patients, from brain imaging studies in healthy normals, and from genetic studies in healthy normals and schizophrenics, all of which provide converging evidence that the midbrain dopamine region and the striatum, which is the main part of the basal ganglia, are critical for this kind of probabilistic category learning. So this leads to the next question: how do rewards and errors chain back in sequence? And by chain back in sequence, I'm referring to the following question. So you have here on the left a mad scientist who's working late in the lab. And the question is, why does he do this? Why are you here? And of course, back in the university, they don't have free food in the cafeteria. So why is he working late? Well, one way to understand it is to look at the chain of reward. So the scientist works late in the lab, then his work leads to publications in science. This leads to a lot of grants from NIH and NSF, which allows him to pay himself summer salary, which allows him to buy a sexy car, which drives the girls wild, which in turn leads to the primary reward. So we see here that there's a chaining of reward from behavior to reward through many intermediate steps. And if we go back to some of these studies from monkeys and these dopamine cells, we see very much the same sort of thing. This is the same type of recording I showed you before of monkeys who are being trained that a cue predicts juice. And what you see is when there's an unexpected juice, the cells fire in response to the reward. But when a cue predicts the juice, as we said before, the dopamine cells no longer fire to the juice, but now they fire to the cue. So now the cue has this sort of reinforcing property that the juice used to. And if you have another cue, Q2, which predicts the Q1, then you see the dopamine activity goes earlier and earlier. So what happens is you get this chaining backwards of reward through a sequence of cues which ultimately lead to some primary reward. So we asked, will we then expect to see similar involvement of the basal ganglia in humans, in how they learn to sort of chain rules, chain cues to get to reward? And the way we've studied that is in a task where we train people to make their way through a house and get outside to get to the gold reward. So you would be here, for example, if you were in an experiment, you'd be in a room that has a blue, red, and a white doors. You would learn to press the blue door. You'd exit from there to another room, which has a green, yellow, and pink door. Once you're in that room, you then press the pink door, and that gets you out to the gold. So you've learned this chain of responses in each room to get from one to the next. And the way in which we train people is we start off with the simplest of training them to go from one room to know which door leads to the reward. Once they've learned that, we bring them back a level and train them for another door, and all the way back till by phase 4, they've learned their way from room to room to room to room with 3 sets of rules to get out. And what we found is when we test Parkinson's patients, by de novo, this means patients who've never been medicated, so they've just presented with the disease but are not yet put on medication, that as the length of the chain increases and it becomes critical to have sort of a sequence of these rewards, that these Parkinson's patients are very significantly impaired, which is what we expected, compared to controls who are able to do the task quite well. Again, genetics provides another way of understanding how the different brain regions are involved in this kind of learning. If we look at people with these are healthy, normal people who have a particular genetic variant, a gene that is a risk factor for Parkinson's, but are otherwise healthy, those people who have the risk factor show the same qualitative deficit. They're impaired at learning. They're more impaired at learning this chain of sequences. And this is a gene that's known to be involved in dopamine production as well. So a brief summary, we can see that behavioral studies in Parkinson's patients, genetic studies in normals, and another study I didn't talk about here, all suggest that dopamine plays a critical role in sort of this guiding, the sequence learning of how we chain back from rewards. So is this learning, the next question is this learning really selective for feedback learning, or is the basal ganglia involved in all forms of learning? So people don't like the weather prediction task. It gets a little boring. So we have another version, which is the Mr. Potato Head task, where we have Mr. Potato Heads who have different features, hat, glasses, mustache, and bow tie, each of which has a probabilistic relationship to an ice cream flavor. And it's the same basic structural task, but you're basically playing a Baskin-Robbins counter clerk trying to predict whether a customer is going to ask for vanilla or chocolate ice cream. And it proceeds the same way. You see a customer, you ask which flavor does he want. You say he looks like a vanilla lover, and in fact, you get positive feedback. And you learn otherwise just the same. And on this kind of learning, we see the same sort of incremental learning over blocks of trials, that when we train people, they get better and better, showing higher and higher % correct responses. So that's the feedback learning that we've talked about so far. Another way is just to stand outside Baskin-Robbins and watch the people come out and just look at what kind of ice cream cone they're eating. And we see here, so we train people that way. They just look at these people with the ice cream cones. They press next to see another customer, and they go through like that, and there's no response required. At the very end of this sort of observational training, because we're not collecting any data, then we ask them to classify people, classify the Mr. Potato Heads as yellow, vanilla or chocolate. And what we see is, regardless of how they're trained, whether they're trained on feedback, whether they're trained by just observational, they show the same overall performance on a transfer task to classify them. So behaviorally, it looks like the learning is the same. But the question is, are these people really learning this the same way, even if their performance is the same? And we know from the past studies that the feedback learning uses the basal ganglia. The question is, what other brain region is involved in this observational learning? And what we found is we compared these two types in a brain imaging study. We compared the feedback learning and the observational learning and asked which brain regions are differentially active in one versus the other. And as we expected, the striatum, which is the main part of the basal ganglia, is more active in feedback training, consistent with what we'd seen before. But the hippocampus, this other brain region that I've mentioned, and the prefrontal cortex are more active in observational learning. So even though we see the same overall behavioral performance, it seems like they're being learned by two different brain regions. So we've noted that since the observational training recruits the hippocampal region, not the basal ganglia, then, and Parkinson's patients have damage only to the basal ganglia, not the hippocampus, you might expect then that Parkinson's patients should be able to learn this categorization if we encourage them to use their hippocampus by presenting the information in an observational framework rather than a feedback. And what we found when we replicated the study with patients is that, as we've seen before, Parkinson's patients are impaired, the PD patients are impaired at feedback learning, just like with the weather prediction task. But when we present them with the observational training, the deficit is remediated. So it suggests one way of remediating some of these deficits by training people in ways that recruit other brain regions that aren't damaged by the disease. So in summary, the brain imaging suggests that feedback learning recruits the striatum, the basal ganglia, observational learning, the hippocampus and the prefrontal cortex, and that in Parkinson's patients, we see consistently that feedback learning is impaired and observational learning is okay. So that's brings me sort of the end of the sort of the basal ganglia part. It's taking about 20 minutes. So now what I want to do is shift down to the hippocampus, which we've argued is involved in developing new representations of the world. So is there more to category learning and how we learn about concepts than just error correction? Or as Gary Larson would say, stimulus, response, don't you ever think? And back in the 50s, a little bit of sort of history of psychology here, when there was this big movement of behaviorism and studies of associative learning, there was a rebellious behaviorist, sort of the origins of cognitive psychology, and Edward Tolman. And he said, well, not all learning is going on because you're giving the animals juice or you're presenting them with food, that animals are intrinsically motivated to learn about the world. He argued that they form cognitive maps of their environment without any specific learning, and that he argued that even without rewards or punishment, latent learning, the implicit learning about our environment and the regularities in the environment, goes on naturally and automatically without necessarily being reinforced in one way or the other. And one example from an animal paradigm of the kind of learning that he's referring to is called latent inhibition. And in latent inhibition, there are two groups of animals. One group of animal just sort of sits in phase one, nothing happens, and then is trained that the cue predicts the US. The US is the unconditioned stimulus. In psychology parlance, it's something like the food or the shock, the significant thing you're predicting. And these animals will sort of just learn normally. And he compared them to an animal that had been pre-exposed to the cue. So in phase one, the cue, which might be, say, a tone, is just presented randomly. The cue occurs every now and then, the tone occurs, nothing is followed by it. And then in the phase two, the animal tries to learn that cue predicts the outcome, and it's much slower. It's as if in that first phase, when it was exposed to that cue in the environment without predicting anything, the animal learned that this cue was irrelevant. And therefore, as you see at the bottom, that animals that are exposed make many fewer correct responses in phase two. They're much slower to learn. And this is not accounted for by the error correction learning, because if you think about phase one, that cue is a neutral cue. It predicts nothing. Nothing occurs. So the error correction notion of learning doesn't account for whatever's happening in phase one, where the animal is learning to tune out this irrelevant cue, and in such a way that it's slower later to learn about it. So what's missing from the Rescorla-Wagner model, this error correction principle? What's missing is latent learning, learning without responses or rewards, such as latent inhibition, or observational learning, standing outside the Baskin and Robbins and just learning by watching people walk out, as well as what Tolman described as learning the cognitive or the psychological maps of the world, our ability to sort of automatically understand our environment and try to map it internally. And what's particularly interesting about these, what's missing from this sort of error correction principle is the fact that these same behaviors are also missing in animals that have damage to the hippocampal region. So if you take an animal and you lesion the hippocampus, or as I'll show in a bit, if you have a human who has damage to the hippocampus, these sorts of behaviors that are beyond the scope of error correction learning disappear. So it suggests that this hippocampal region may well be critically involved in this sort of latent learning that Tolman had talked about. So if you take an animal and you remove the hippocampus, simple acquisition is normal. The animal can learn, a cue predicts an outcome. But if you do something like latent inhibition, what you see is that the control data is what you just saw a minute ago, but animals with hippocampal damage don't show that effect. In fact, if you compare the exposed condition, the animals that have been pre-exposed to the cue in phase 1, you'll notice that the hippocampal lesioned animals are learning faster and better than the normal animals. So it's actually a very sort of unique case where the brain damage is actually improving performance in this case, because normal people tend to tune out these cues and have trouble learning from them. Animals with hippocampal damage don't tune it out, so they're actually faster to learn. So you might think, well, that it's going back to acquisition, sort of learning simple cues. For a long time, people thought that, well, the hippocampus isn't involved when you're learning sort of a simple association. Tone predicts shock or something like that. But in fact, even though there's no effect of a hippocampal lesion on these animals, if you look inside the brain, the hippocampus, in particular, is showing lots of activity which models the response, which looks just like the behavioral response, in this case, a blink to a cue. So it suggests a sort of a caveat that even though a brain region is not needed, it clearly participates in even the simplest form of learning. And the question that we pose is, what is it doing? It's clearly involved in this latent learning. It's doing something in the simple acquisition, even if it's not evident in just the trials to criterion learning. And so let me give, before I sort of talk about some of our thoughts about what the hippocampus does, let me give a brief interim summary. Latent learning is learning about the world without explicit consequences, such as reward or punishment. One example is latent inhibition, this task I talked about where there's pre-exposing to a cue. Latent learning is not explained by the Rescorla-Wagner model of error correction. But hippocampal region damage impairs latent learning, which suggests that something about latent learning, learning from the environment, learning from cues you're just exposed to, is critically involved with hippocampal function. And finally, the caveat that the hippocampus isn't necessary for simple acquisition, but it's clearly involved somehow. So what does the hippocampus do? This has been for many years the $64,000 question in the neurobiology of learning and memory. What does this brain region do? Everybody knows it's important for learning and memory, but to actually pin down what is its algorithmic function in the brain has been difficult. And so in psychology, when we're faced with a problem that troubles us, we remind ourselves WWWJD, which is a reminder to think back to our ancestors and ask ourselves, what would William James do? So William James was the father of American psychology. He was the first professor of psychology at Harvard University, and he wrote the first textbook of psychology, An Introduction to Psychology. And in this textbook, he has the following quote. He says, "If all cold things were wet and wet things cold, if all hard things pricked our finger and none others did so, would we come to distinguish between coldness and wetness and hardness and pungency, respectively?" And what James was saying in this paragraph was that there are many things in the world that tend to co-occur. Coldness and wetness. There are a lot of things that are cold and wet. There are a lot of things that are hard and pungent, what we now call sharp, that co-occur. And he was arguing that when these features in the world tend to co-occur, we have a tendency to cluster them together, to treat them as some unitary whole. And it's a principle we've described as redundancy compression. Redundant in an informational sense, when two cues are informationally redundant, we tend to compress them and treat them in a unitized fashion. James also described another experiment that he did at Harvard back in the 1800s, where he initially was not much of a wine expert, and claret and burgundy to him tasted just the same. And so he decided to train himself to see if he could distinguish them. And so he sat with a bottle of one and a bottle of the other. And although initially they tasted the same, as he drank from one glass and then the other glass, then the other glass and the other glass, and so on and so forth, before he passed out, he began to sort of extract out the sense of what it was that made the distinguishing between a claret and a burgundy. And this is a principle which one can think of as predictive differentiation, that initially when things may seem very similar, that if they're mapped to different outcomes, or in this case, they're mapped to different bottles, or the labels on the bottles, that you learn to distinguish them in such a way that ultimately they seem much less similar. And we call this predictive differentiation. So the idea is that when a cue predicts two different outcomes, we'll tend to differentiate these cues. So where does this occur? Where does redundancy compression? So this was James described this over 100 years ago as being a psychological theory of how our understanding or representation of the world changes. Where does this occur in the brain? So one way is to do what I would call a brain imagining study, is I put myself in a magnet and asked, what is it, where can you see redundancy compression and predictive differentiation in the mind of a New Yorker like me? And this is what a New Yorker's brain looks like, a New Yorker's map of the world. And there are a couple of things that one should note about a New Yorker's view of the world. That if you look up in what we call the dorsal region of the map, you'll see redundancy compression. So New Yorkers tend to view all states in the Midwest that begin with the letter I as being functionally equivalent. On the other hand, if you look down in what we call the ventral region, you see predictive differentiation. We know that the best Thai restaurants are on 9th Avenue and there's nothing on 10th Avenue. And so we tend to expand out this region of our brain. And so this suggests that the hippocampal region, we've argued, is critical for these sorts of distorted maps, that they create mental representations that expand representations that need to be differentiated and compress over those that are functionally equivalent. And the way in which we've argued that this takes place in the brain is through an interaction between two different brain regions. The hippocampus, which is monitoring the world in this sort of latent fashion, taking all the information in that comes in, and developing new representations that look a bit like that New Yorker cover, which are biased by the regularities in the environment, the redundancy compression, and the predictive differentiation, and that those are then adopted by other brain regions. And in one sense, from a psychological perspective, one can see this as sort of mapping two different traditions into two different brain regions. That the cortex and the cerebellum, where long-term memories are stored, are capturing the sort of learning that Aristotle talked about in Hull and Rescorla and Wagner, the sort of learning of these associations of stimuli to outcomes and behaviors. And that the hippocampal region is capturing more what Tolman was talking about when he talked about latent learning, and James with these changes. That is sort of how we develop our representations of the world and how we alter what's similar to what. And that they work together. And how can these be instantiated in the brain? I want to give sort of just a very brief sort of characterization of some of the kinds of network models that one can develop that capture these principles. And this is not necessarily how the brain explicitly does these sorts of computations, but it suggests a way in which a complex associative network can begin to develop associations and learn mappings using these principles. So a very simple model which we developed suggested that one could think about the long-term memory, the cerebellum or other brain regions, as a simple feed-forward network of associations from the stimuli to the output. And that what the hippocampus is characterized as is what we call an autoencoder. It takes everything in the world and maps it to what's essentially the next step of everything in the world. So all the cues mapping to all the cues plus the prediction of the US, the sort of the critical thing we're trying to predict, the food or the reward or the shock. And that it does so through a narrow channel. So essentially what this is doing is sort of similar to what happens when you do a compression of a data file. So if it's compressing out the redundancy through this narrow channel, it's developing a new representation. But because this representation has to be able to predict the next step, the next relevant outcomes, it's compressed, except it also has to be differentiated based on the things that are relevant for you to differentiate. And what we argued was that this sort of kind of architecture, this what's called an autoencoder, develops representations which are compressed and predictive, and that other brain regions use this. And what's critical for this sort of learning is novelty detection. In other words, that the system is constantly sort of building a map of the world through this network on the right. And when something comes in that it doesn't recognize, it encodes it and decodes it in this two-layer network. And if it doesn't match, it says we need to adjust the weights because there's something new and different in the world. What happens when you remove the hippocampal region in an animal or a person, and I'll talk about the latter in a second, what you get is basically a very simple error correction network left that just uses whatever the fixed representations that it had originally, and this approximates the sort of Rescorla-Wagner error correction learning that I talked about a minute ago. So that's a sort of very brief overview of one kind of sort of connectionist architecture that gives you that. And we showed that this model is able, we would compare the intact model with the lesion model to the data. And as you saw before, we see the model accounts both for the fact that simple acquisition is unimpaired by the lesions, but in latent inhibition, you see a facilitation that the lesion in the lesion model is actually faster to learn in phase 2. And the reason it's faster to learn is that what's happening in phase 1 when the model is exposed to the cue is it's not just exposed to the cue, it's exposed to the background context, what in the animal experiment would be the box and the lights in the room. And because the cue and the context are co-occurring and they're redundant, they tend to be compressed together. So what you're actually seeing in the model is that it's beginning to treat that cue as part of the context, to compress it all down together and treat it all as this sort of unitary, irrelevant context. And so why learning is slow in phase 2 is that the network has to reorganize the representation so as to extract out the cue and differentiate it from the background context. And it's that necessity of representing the information that slows down the learning in the intact model and explains why the intact model and intact animals are faster to learn, are slower to learn. And for those who are interested in learning more, we have a book which Eddie referred to, which describes in more detail some of these kinds of theories of the hippocampus. So, as interim summary, the hippocampal region is involved in developing mental representations that reflect regularities in the world, biased by redundancy compression and predictive differentiation. These are then used by other brain regions that store long-term memories. And the loss of the hippocampal region is associated with learning that takes place with non-adaptive stimulus representation, sort of a rigid view of the world. So what are the implications of this for people with hippocampal damage? One way we get people with hippocampal damage is people who've had an anoxic episode. They've lost oxygen to the brain, and this basically destroys the hippocampus if it doesn't kill them. And we study these people. These are people like, you saw the movie Memento, the sort of someone who's amnesic. All their old memories are intact, but they can't learn new information and new facts. And we train them in 2 different types of experiments. All the experiments have 2 phases, one in which they're exposed to some regularity, where we expect that that a normal person would be developing a representation that encodes that regularity. And then we either have a positive transfer, where they have to learn something that uses the same regularities, so the phase 1 should be helpful, or we shift it so that there's a different regularity going on they need to learn, and if they were exposed to the regularity early on and they learned it, that would mess them up a bit. And what's particularly interesting is these negative situations like latent inhibition, because it suggests that the brain damage should actually facilitate learning, because you're giving the brain damaged person an exit from having a mistaken reorganization that the normal person will do. So learned irrelevance is a paradigm which is basically the same as latent inhibition, except that early in training, you're exposing people not just to the cue, but to the cue and the outcome, the CS and the US, but they're uncorrelated. So a tone might occur occasionally, a shock might occur occasionally, but they're not paired together in any way in phase 1. And then in phase 2, the tone is paired with the shock, and you look at how long it takes an animal to learn. Tone predicts shock. So the human version of this involves training people with a task we call Merlin. Can you predict whether there's a rabbit under the hat? And you see these trials, and in this case there is a rabbit under the hat, and the question is, how do you learn to predict that? And the way in which we do the study is there's a phase 1, the exposed group, similar to the animals that have seen the cues uncorrelated. There's a word, a magic word, in this case blay, which tells you whenever the magician says blay, there's going to be a rabbit under the hat. And all other words, there's no rabbit. And the colors of the balloons vary and are irrelevant. And then in phase 2, the colors become relevant. And in fact, there's now a rabbit under the hat whenever the balloon is red. And we compare that to people who get the same phase 2, but they're not exposed to the color in phase 1. They're learning that blay predicts the rabbit, but there's no color. So it's functionally the same as a situation with the animals, where you're either exposed to a cue or not exposed to a cue. and then you have to learn about that cue. And what we expect is that healthy controls should show this learned irrelevance effect and learn more slowly in phase 2. And that's exactly what we see. That in this case, we're looking at errors, that people who are exposed in the exposed condition make many more errors in phase 2. On the other hand, amnesics are learned faster than the exposed controls in the exposed condition. So we see here that they're doing better. Again, like in the animal conditions, these amnesic patients who are overall massively impaired are doing faster in the phase 2, and they're completely normal on the phase 1. Another way of looking at some of the implications of these representational changes in the brain is a situation where there's a positive transfer, where something that's relevant in phase 1 applies to phase 2. We have a task that's similar to the card games that they play in the city, where you're trying to figure out where's there a peanut, in this case where there's a smiley face. And the smiley face you might guess left, and in fact it is underneath the red octagon. And we train people on many different pairs like this. And in some of the pairs, there's a color rule, so that there's a red versus yellow, and the shape is irrelevant, it's constant. And in others, there's a shape is relevant. So in this case, green square beats green triangle, and the color is constant and irrelevant. And we have 4 of each. And then later on, we shift them in phase 2, once they've learned phase 1, to this, which they've never seen before. And given that you've never seen it before, you might just guess randomly. Or you might have extracted the relevant rule from phase 1, which said that red beats yellow. In which case, if you had extracted that relevant rule, you would guess correctly. So what we basically have here is concurrent discriminations with the rule. And the rule stays the same from phase 1 to phase 2, but the irrelevant aspects change. And what we're looking at is, are people, when they're learning in phase 1, are they just learning about the specific items that's good enough to solve phase 1, or are they extracting the general principle that allows them to apply it in a broader range of situations? And we expect that the hippocampus is critical for this, that it's critical for developing these appropriate representations. And so what we see here in these amnesic patients is that both the controls and the amnesics are perfectly fine during phase 1. They're able to do simple learning. But in phase 2, the amnesics make many more errors. It's as if they're starting over from scratch. And although they still remember the discriminations from phase 1, which we test otherwise, but it shows that however they learn phase 1, although it seems normal, they haven't learned it in a way that allows them to apply that knowledge more generally when some of the features change. Another population that has hippocampal damage is people who are on the route to having Alzheimer's. So before you actually have Alzheimer's, you can see in people the beginning of shrinkage of the hippocampus. And it's a very mild degree of shrinkage. And in fact, if you test people neuropsychologically with memory tasks, they look normal. And our question, which had a lot of clinical relevance, was, could we develop a behavioral task which picks out these people who, from imaging, we know have the beginning shrinking of a hippocampus, which is a sort of a warning sign of getting Alzheimer's, but before they actually show any behavioral deficits on standard neuropsychological assessments. So here's a standard neuropsychological assessment. This is just read a paragraph, 5 minutes later, ask people if they remember the facts. And you can see that people who are these elderly who have a little bit of hippocampal atrophy, that's the yellow, the HA hippocampal atrophy, they show no deficit on this delayed paragraph recall. On the other hand, when we take this same group and we do it on our discrimination transfer task, what we see is that qualitatively these people who are completely normal behaviorally look just like our amnesics, that they learn fine, but they show a significant impairment when it comes to the transfer. So what this suggests is that if you lose your hippocampus, all of it, there are these massive deficits in learning new information that are obvious. But if you have just a little bit of damage, that the first thing that you start to see are these generalization deficits. And we've done some other work. We followed up these people 5 and sometimes 8 years later. And what we found is that the people who did poorly on our transfer task, independent of how they were sort of characterized clinically, are the ones who are the most likely to convert actually I take it back, this is 2 years later, most likely to convert 2 years later to either MCI, which is sort of a mild version of Alzheimer's, or to Alzheimer's itself. So it suggests that these sorts of behavioral measures might actually have a clinical relevance in predicting cognitive decline. The sequence learning task I described to you before, when we take a population of people who have AMCI, so AMCI are people in this early sort of pre-Alzheimer's stage who are showing amnesic deficits, they're showing memory loss in this case, They're obviously people who've come into a clinic because of this memory loss. They're very likely to get Alzheimer's in the next few years. They're totally normal on this task. So that's sort of one of the things interesting is that even though these people are much more demented than the de novo Parkinson's patients, they're unimpaired at learning the sequence task. On the other hand, we did a transfer task, which I didn't mention before, which is after you do the sequence learning task, we gave them the same sequence, but we changed around the distractor doors. And in fact, in some of the rooms, we put one door, which was a correct door, in another room, and as well as a totally incorrect one. So we changed around the context of the distractors. And then what we found is that the Parkinson's patients who had been very impaired at learning, they weren't affected at all by this. But the amnestic MCI patients, these sort of pre-Alzheimer patients, who were completely normal at learning it, they broke down on the transfer. So we see that same pattern, that damage to the basal ganglia slows down learning, but doesn't impair your ability to use the learning once you get it. Damage to the hippocampus will not impair simple learning, but we argue that you're learning it in a dysfunctional way that doesn't generalize. So an interim summary here. As predicted by our model, you see these transfer generalization deficits on this learned irrelevance, the Merlin task, on the concurrent discrimination task, on the acquired equivalence task. And the question is, why do you see these deficits on transfer generalization? And we've argued that the reason you see the deficit in phase 2 isn't because of a problem in phase 2, isn't because of a problem going on in the generalization. It's because of a problem that occurred back in phase 1 when people were learning these tasks, but they were learning it in a way that didn't have input from the hippocampus and therefore was dysfunctional in some way. And let me give you an intuitive understanding of what's going on in our model that provides the interpretation for this. So the question is, how does the hippocampus improve generalization? And in our model, the hippocampus is essentially providing a support role. It's developing the representations that are used to create the rules that are used by other brain regions. So one way of thinking about a brain network or a simple mathematical model of a network is in terms of the state space. So every possible state of this network is a highly dimensional space in terms of all the possible different weights there could be on every single link. We'll simplify and think of a two-dimensional space here. And so every possible state of this network is a point in the space. When the network begins, it has some initial status there. And learning in this geometric representation is simply moving from one point in the state space to another. As you adjust the weights, as you change the rates, you move to another point in the space. And so learning is getting from a place where you're not performing well to finding a point in the space where you are able to perform. In most of the kinds of tasks, because you have such a high dimensional space of possible solutions in these networks in the brain, the training problems under-constrain the possible solutions. There isn't just one network solution in most cases. There's a whole subregion of the space that will solve the training task because the training environment under-constrains the possible solutions. And what a network will do as it's trying to reduce the error is it will basically find the closest point in the solution space, and it will move in that direction directly. And as soon as it gets to one that solves it, it will stop because it's doing this sort of error correction learning. And once it corrects the error, anyone will do. So learning without a hippocampus, we've argued, is basically doing what network theorists call gradient descent. It's moving to the closest part of the solution space. However, that part of the solution space, there's a subset of that solution space for the training, which is the solution space which is, one could think of as the most parsimonious, the solution space which not only solves the training task, but is the most likely to generalize to a broader range of situations that haven't been experienced yet. And so what we've argued is that what happens with the hippocampus is the hippocampus actually makes it harder to learn because it says it's not enough just to get to the solution space and solve it. But you have to sort of dig deeper and find the region of the space that's going to have other constraints. And in particular, there's going to be a constraint that the representations used to solve it have to satisfy these constraints of redundancy, compression, and predictive differentiation. So basically, those constraints, redundancy, compression, predictive differentiation, are added to the problem in training, and they therefore constrain further the set of optimal solutions. And what we've sort of argued in these experiments that I've shown before is that without the hippocampus, you can learn fine, but you don't transfer well because you've found a suboptimal region of the solution space for the training task. So let me just skip here and talk a little bit about how do some of these ideas map on to some applied issues, some problems of machine learning, because I understand there's a lot of interest here in machine learning. So we've done a bunch of work over the years with the military, with the Navy, and with DARPA, asking how is it that we can take these brain systems for pattern recognition, for novelty detection, for classification, and begin to apply them to real-world problems. So one we've worked on is sonar classification. We have a lot of data on sonar signals that have come back from rocks versus mines, different categories, from biological signals versus submarines. And the question is, how do you sort of classify them into the different categories? Obviously, a problem of relevance to the Navy. And this is not very high contrast. I don't think I'm going to use green anymore here. So can people see that graph there? So what I'm graphing here is we have a body of data that's categorized. We start with 25% of the database, and we train it using this BPNet is a simple backpropagation network, a simple feed-forward network. And what we're graphing is overtraining the mean squared error, which is sort of a measure of the global fitness. The smaller the error, the better it's doing on all the possible sonar signals. And what you see in a typical graph like this is across training. This is 5,000 blocks. The error graph goes down. That's exactly what you expect. But the problem is, at each point in training, we said, well, at the network at this point, how is it doing on the other 75% of the sonar signals that it's not being trained on? And what you see here is, in fact, it's actually getting worse. And this is it starts out here, and as you train, it gets worse and worse. And this is a phenomena that's familiar to statisticians known as overfitting. That what's happening is that even as it's getting better on the training signals, the error is increasing on the generalization because it's overfitting the training data. It's picking up more and more of the idiosyncrasies of this small subset of the data, and it's making it actually worse to generalize. In comparison, we looked at our cortico-hippocampal network, which is very similar to this backpropagation network, except that it's constraining the solutions the internal representations so that they are additionally constrained by redundancy compression and predictive differentiation. As you see, much like we saw with the patients, there's no difference for sort of the simple learning. Okay, the error graph looks the same. But the difference comes when you look at the transfer generalization. That what's happening here is as you continue to train on the 25% of the training sample, that you're getting better and better and better at transfer, and significantly better than with the simple network. So what we see here in an engineering application is the same principle we saw with these amnesic and Alzheimer patients, which is that adding these hippocampal constraints is similar, in some sense, to, just to put it into the local vernacular, you can think of the hippocampus then as determining the page rank of different solutions, that there are a number of different possible relevant solutions. And rather than just sort of coming up with any number of ones that are going to work, the hippocampal region is creating a metric of sort of optimal solutions, given what's the input for training. And that metric is helping it identify the subset that are going to be of most relevance and most interest when you come to generalize to something new. So that's one application. Another application is the problem of novelty detection. I mentioned that the way in which our system works is it uses this autoencoder, this sort of mapping from the input to a compressed representation and back out to the input, and develops these representations. And for any time there's a stimulus that it doesn't recognize, the encoding and the decoding are not going to match. And you'll get a mismatch between the input and the output of this network. And that's a measure of novelty. So the error, the mean squared error on these output nodes is a measure of the novelty of a stimulus. And the question of determining novelty has a wide range of applications. For example, if you're flying a helicopter and the gearbox is making a funny vibration, that's something you want to know about. You don't really care why it's vibrating. You just know that if this is a vibration that's never made before, this is not a good thing. And there are a lot of problems where people generally often think about sort of pattern recognition, but novelty detection is a very relevant domain where it may be very hard to get examples of novel systems. So it's very hard to collect data on how a helicopter gearbox vibrates when you're flying it. Okay? You can collect a lot of data on good helicopter gearboxes. That's easy. But not many pilots are going to want to sort of fly a helicopter that's impaired just to collect data for your neural network thesis. So one of the things we did is we worked with the Navy on the actually, this is a problem with submarines. I'll come back to the helicopter in a minute, where we're looking at the pumps on submarines, collecting data on training them. So we took all the submarines, the good pumps, And we trained it so that ultimately there was sort of on the training that these are all sort of no-fault pumps, good working pumps. And then what we did is we looked at test pumps that were either faulty or no faulty. And what we found is that the faulty pumps generated a much higher novelty score in our hippocampal network. So the idea being that you train the system to sort of recognize the environment of a healthy pump, and then when you present it with something that's not healthy, it's aberrant in some way, you don't really care what way, but its novelty signal is different. So you can use these sorts of autoencoders as a way to build effectively statistical models. And we've also done a lot of work with the Navy with helicopters. And in fact, we had an incident where the Navy had sent us a whole bunch of data, including what they said were good training helicopters, good training data from recently rebuilt gearboxes on the CH-46 helicopter. And one of the databases just looked way out. It was a big outlier from all of the rest. And so we sent a message back to the 7th Fleet in the Pacific, who had sent us this, saying, we have a problem with this particular helicopter data. It doesn't look very healthy. And they said, no, that was just rebuilt 3 months ago back in San Diego. But nobody wanted to be the guy who sort of ignored it. So they took it. It was actually out in the Pacific at Fleet Maneuvers. They took the gearbox out. They shipped it back to San Diego. And sure enough, when they had rebuilt the engine, someone had screwed in one of the parts wrong, which ultimately would have been a catastrophic fault if it had happened at sea. So these are the kinds of applications where you can see that novelty detection plays a way of sort of building a model of your environment and recognizing when something goes wrong. So that brings me to the end. Eddie mentioned there's a book. We have a book that's actually coming out this next month. If anyone's interested in understanding the brain mechanisms of learning and memory, models of learning and memory, and how they break down, this is a new book that'll just be out. So thank you. That brings me to the end. So I'd love to hear from all of you, particularly, not only sort of general questions, but also these sorts of applications of machine learning and generalization and novelty detection. Are there any sort of problems, that you're working on here for which those might be relevant approaches?
No, and I'll give you the mic.
Yeah, I gotta.
Hi. I was wondering if you could elaborate on the link between your auto encoder and the backprop network. It looked like there was a line, but I couldn't tell.
Sure. I just skimmed over that. In fact, I usually skip over those slides, but for Google, I put those slides back in. So the way the model works is that you have this autoencoder that's developing these new representations. And then those new representations become the training signal for the representations in the backprop net. So basically what's happening is the backprop net is learning to mimic. Actually, it's learning some linear transformation of them. So that basically the hidden units from the autoencoder become the training signals for the hidden units in the multi-layer network. So what's happening in this long-term memory network is it looks like a typical backprop net, but in fact the two layers are being trained independently. That the bottom layer of weights are being trained by input from the hippocampal region, which is telling it how to represent it, and the upper layer of weights are being trained by the environmental feedback, and that's your standard error-correcting feedback.
So when you present training data, do you
Sorry,
present...
I can't hear you with all the noise out there.
Sorry. When you present training data, do you train the autoencoder in a block and then train the backprop net, or is it all...
No, everything's being trained simultaneously.
Okay.
So what's basically happening is that in the intact system is the long-term memory network is learning while the representations are evolving, which is one of the reasons why the intact model takes longer to learn, because its representation is moving around. And in fact, one of the things you see in animal literature is that although the hippocampus is needed for complex tasks, especially nonlinear tasks like the exclusive OR, that on simple tasks, hippocampal lesion animals do better. And part of the reason we've argued is that it's because an intact animal is learning more than just the training task. It's learning to develop this appropriate representation, and that representation is evolving during the early stages of learning.
If I could just
Sure,
comment.
sure.
I was asking because I was wondering if you had looked at sleep and the role of sleep in all of this, because it seems
absolutely,
like it could be
absolutely.
interesting.
So we have some studies that are just about to come out. We've been working with Bob Stickgold at Harvard Sleep Lab, and what we've been doing is, I actually have the data here, but never mind. I'll just describe it. So what we've been doing is we've been training people on these tasks either at 9 a.m. and then testing them again at 9 p.m. or training them at 9 p.m. and 9 a.m. and asking what is the effect of sleep on this learning. And there's been some past work by Bob Stickgold that's argued that REM sleep is critical for hippocampal-dependent learning, but not basal ganglia-dependent learning. So what we expected then, if that was the case here, we expected that feedback learning that we described should not be improved by overnight sleep, but observational learning, which we argued is hippocampal-dependent, should. And that's exactly what we found, is that when we test people with overnight sleep, that the next morning, those who did the observational training have actually shown an overnight improvement, while those who did the feedback training did not. Except if we train people for just a few trials. So you saw before I talked about how I showed briefly that this basal ganglia activity increases during learning, but that even in feedback training, there's sort of an early phase of hippocampal activity. So we train people just on the early phase, just enough trials so they presumably still have a very strong hippocampal component. In the model, we'd argue that's because the representations are still being formed, that early hippocampal activity is the developing of the representations. And those people trained on just a small amount of feedback training show an improvement overnight. So in both cases, we see that aspects of learning that recruit the hippocampus are improved by sleep. And moreover, when they use the hippocampus, you see more REM sleep the next night. So in fact, there's a correlation between how well people do and the percent of REM sleep. The percent of REM sleep after they did the training. So the REM sleep is essentially an index of how much you have to encode in memory. And so it suggests a sleep system which has this homeostatic control that you'll get more REM sleep if you have more that you've learned using your hippocampus that you need to encode. That was probably much more than you wanted to know about sleep and memory.
That's really interesting.
But that's a whole line of work that we're doing. And ultimately, of course, the question is, can we come back to the models and begin to incorporate some of this consolidation in sleep?
It seems like there's another group coming in here. So Mark and I are going to be having lunch out in front of Charlie's afterward. If anyone wants to join us, please come by. And thank you very much.
Great. Thank you.